traditional view and say that the function of the nucleus is to control cellular activity. So really it does that, right, by holding on to DNA, but DNA is really just a blueprint or a pattern for proteins. And so the way that it's controlling cellular activity is by producing proteins at the appropriate time. The structure is interesting. So the nucleus consists of I think I'm going to draw it over here. The nucleus consists of 2 layers of plasma membrane. So that would look like this, right? We would have two layers of phospholipid bilayer. So the cell, right, is surrounded by a single layer of plasma membrane. The nucleus is surrounded by 2 layers of plasma membrane, which really increases how protective that membrane is, right? I mean, it's super protective. The problem is we need to still communicate with our DNA. Chemical signals need to come into the nucleus, chemical signals need to leave the nucleus. So the way that that's accomplished is with protein channels that span both layers of plasma membrane. So that is called a pore. So there are these pores that are just kind of speckled throughout the surface of the nucleus that allow chemical signals in and allow important stuff to leave the nucleus. So if we were to describe the structure of the nucleus, it's 2 layers of plasma membrane . That are connected by protein channels. Okay, so the nucleus's job is to control cellular activity. That's like the ultimate goal, right? The proximate goal is to keep the DNA safe. What that means is when we want to replicate a DNA, or excuse me, when we want to replicate a protein or make a protein from our DNA, all of the stuff to make the proteins exists outside the nucleus. So we have 2 options. One, we can send our DNA out to the cellular machinery, or we can make a copy of our DNA and send a copy of the DNA out. And what we opt for is option number 2. So we make a copy of our DNA, send that outside the nucleus, and that copy goes and finds the right cellular machinery to make the protein. And that copy of our DNA is called RNA. That's ribonucleic acid. So we make an RNA copy of our DNA, and that RNA copy is read by a ribosome, and a protein is made from it. So the DNA is a super long molecule, as I mentioned earlier. And there's protein scaffolding associated with the DNA molecule. We've taken that DNA molecule and we've chopped it up into discrete regions called chromosomes. And we used to believe that the number of chromosomes indicated the complexity of an organism, but we know that it doesn't. It varies greatly between organisms. But what that allows us to do is to take specific regions of our DNA that are important to those cells and express them. Or regions of our DNA that aren't important to those cells and compartmentalize that DNA really, really tightly. We can take that DNA and we can wind it up so it's completely inaccessible to cellular machinery. For example, your eyes are really, really good at seeing things, right? And you would want the cells in your eyes to create the proteins necessary to receive light and to transmit those messages to a central neuron system. However, you don't necessarily want your eyes to breathe for you, right? And you don't want your eyeballs to grab stuff. And so the regions of DNA that are going to be responsible for like muscular protein production or respiration are going to be compartmentalized and inaccessible. Does that make sense? That's the value of chromosomes. We can make specific regions of the DNA accessible to cellular machinery so that those cells can do their jobs. And we can compartmentalize the stuff that isn't all that important to those cells. When the DNA is completely unwound, when it's not compartmentalized, that's called chromatin. We'll talk about chromosome structures a little bit more in chapter something or other when we talk about DNA replication. There's an important region of the DNA called the nucleolus. And so this diagram shows you the nucleus, and then there's this darkened region in here called the nucleolus. It's highly condensed DNA that's responsible for making the RNA that goes into ribosomes. So it makes ribosomal RNA. I like asking questions about the nucleolus on the exam. So again, the nucleolus is a condensed region of DNA within the nucleus that's responsible for making ribosomal RNA. And again, ribosomes are just these crazy little RNA organelles that manufacture proteins. They don't have any protein or any membrane associated with them.
So can you repeat that one more time?
Yeah,
the condensed region
of the nucleus, of DNA within the nucleus, condensed region of DNA within the nucleus that makes ribosomal RNA. Are we okay with the nucleus? So the next structure that we're going to talk about is the endoplasmic reticulum. And the endoplasmic reticulum is, it exists in two parts. There's something called smooth endoplasmic reticulum and something called rough endoplasmic reticulum. So I'm going to give you the structure and function of each, the rough and the smooth, independently. So let's do structure first. Okay, so the structure of the endoplasmic reticulum is just a membrane sac that surrounds the nucleus that's highly convoluted, super wavy, and embedded with ribosomes. And here when we spell sac, it's just SAC. Convoluted sac. Membrane around nucleus. embedded with ribosomes. Now, we've talked about ribosomes a lot. So if the rough endoplasmic reticulum has a bunch of ribosomes, what does it do? Yeah, it makes proteins. That's exactly what it does. And it makes a specific kind of protein. It generally makes proteins that are intended to be secreted from the cell. So the proteins are made inside the cell and then they're released outside the cell. So the function is protein construction and these proteins are secreted from the cell. Okay, so think about your body, and think about where you're making a bunch of proteins that you release outside of a cell. So where would you expect, which tissues would you expect to find a high concentration of rough endoplasmic reticulum? Skin, why the skin?
Because of your sweat and your constant losing of cells and regenerating.
I love it. So the cell cycle, reproducing cells, is really active in the skin. You're absolutely right. And I like the way that you're thinking about secretion, because we do release sweat and pheromones and things like that from our skin. But most of those things aren't protein-based. So where could we be making and secreting a whole bunch of proteins? Yeah. Yeah, the digestive system is a great one. So remember the digestive system, what we're doing is we're taking proteins, enzymes that are going to catalyze the catabolic reaction of digestion. So we're dumping a whole bunch of digestive enzymes into our stomach. They're finding these molecules and breaking them down. So stomach lining, large intestine lining, has lots and lots of rough endoplasmic reticulum. Now there are other ribosomes that exist just kind of floating around in the cytoplasm. Those ribosomes are going to make proteins that remain within the cell. I'll say that again. Ribosomes inside the cytosol that are just floating around in the cytosol make proteins that remain within the cell. That's correct. They're not embedded in the rough endoplasmic reticulum. There's also a smooth endoplasmic reticulum. So the smooth endoplasmic reticulum has the exact same structure as the rough. It just lacks the ribosomes. So the structure is a convoluted sac of membrane.
It just lacks what?
Ribosomes. So before I talk about the function of the smooth endoplasmic reticulum, we need to talk about two unifying features of the endoplasmic reticulum, the rough and the smooth. The first of which is it's a highly convoluted sac, which means it's super wavy. Why do we have super wavy structures?
Surface area.
Surface area, that's exactly it. We're increasing the surface area of these organelles. Now, if you think about it, the more surface area there is in the rough endoplasmic reticulum, the more places there are to have ribosomes, and the more protein production we can accomplish. Secondly, why are they located right by the nucleus? Okay, think about it in terms of distance. As soon as an RNA copy is made within our nucleus, it just goes right out and immediately begins to interact with the ribosome to make a protein. So location of the endoplasmic reticulum is really important, right? It's close to where the source of those chemical messages are being emitted. And then it's convoluted to increase the surface area on which we can do some work. So the function of the smooth endoplasmic reticulum, it also manufactures things, but it manufactures fats and some sugars. But it also detoxifies things. So there's this phenomenon, and I should mention I don't drink, but there's this phenomenon, right, among people that do, that the first time you drink, it's really, really easy to feel the effects of the alcohol. You can get a buzz really, really easily. But the more you drink, the more difficult that buzz is to achieve, right? I'm assuming that's right. That's right, okay. It's a well-documented phenomenon. The question is why? Why would that be the case? And the answer is, when you drink, oh, were you going to answer?
I was just going to say your body builds up tolerance.
Okay, so it's this tolerance idea. And the question is, what's the biological explanation for that tolerance? And the answer is this. When you drink alcohol, alcohol is a poison. And you're poisoning yourself, and that's what makes you feel good, right? And that's, in fact, the case with most narcotics and chemical compounds. Anyways, what happens is you drink a bunch of alcohol, and then alcohol is sent to cells that have a really high concentration of smooth endoplasmic reticulum. And the smooth endoplasmic reticulum is able to take that alcohol compound and break it down so that it no longer has that toxic effect in your body, which is why the buzz eventually goes away. The more you drink, however, the more your body thinks, my goodness, I'm having this toxin dumped into my body with some frequency, I ought to make some more smooth endoplasmic reticulum. So the amount of endoplasmic reticulum generated by those cells increases, and your capacity to tolerate alcohol also increases, which means that you need to drink a little bit more to get the same sorts of physiological effects. Does that make sense? Okay, so with what you know about the body then, which tissues or which organs would you expect to find a very high concentration of smooth endoplasmic reticulum? The liver. The liver is a great one, right? Because that's where our body detoxifies alcohols, which is why cirrhosis of the liver is a problem among those who drink a lot. Yeah, Mac.
So is it true that if someone gets like that part of the cirrhosis from the liver removed, that the liver can regenerate itself, or is it still going to like regenerate the cirrhosis part? So I agree with that.
No, it should grow healthy cells, healthy tissues.
Okay.
Yeah, good question.